Good morning. If you remember one thing from this talk, I hope you remember lots of things, because I think it's a really interesting subject. If you remember one thing from this talk, I want it to be this. I am not a doctor. I'm going to talk about viruses, I'm going to talk about molecular biology, I'm not going to talk about disease. I am absolutely not qualified to diagnose any disease, to advise on treatment, to talk about disease progression, anything like that. I study molecular biology. I am not doctor, nurse, paramedic, pharmacist, anything useful. Please do not make important life decisions like medicine about on the advice of bearded men shouting at you in fields. If that kind of thing appeals to you, I have been thinking of starting a cult. By all means, talk to me about that. Do not try and take medical advice from me. I will do my best not to say anything that sounds like medical advice, but also, please don't read in. Having talked about dogma and cults, we're going to talk a little bit about the central dogma of molecular biology, introduce DNA, RNA, proteins, and talk about the structure of the cell, and then go on and talk a little bit about how viruses get in, exploit the various systems inside the cells. So then, the central dogma of molecular biology. Like all the very best dogmas, it's quite simple. It has some very sort of foundational basis. It's very useful, and it's mostly true, so long as you don't think about it too hard. I'm being a little unfair to Crick there, but it is mostly true. The idea is that we have DNA in the cell as the source of truth, deoxyribonucleic acid. This is an enormously big, by molecular terms, molecule that hangs around in the nucleus of the cell, containing vast quantities of information. DNA is the source of truth, the source of all information in the cell, mostly. The information in the DNA needs to be used somehow. It gets copied into RNA, ribonucleic acid, which is very, very similar, a bit less stable, and we get small regions of DNA copied into RNA. The RNA leaves the nucleus of the cell, and then this is used as a template to make proteins, mostly. But this is close enough to being true, and it's a very good mental model to get started with. So you have DNA copied into RNA. RNA moves and either does something itself or serves as a template to make proteins. And then the proteins in the cell go off and do all sorts of stuff. And as you can see here at the bottom of the diagram, when we say a protein, what we mean is actually a long, stringy molecule. Each of the subunits is called an amino acid. There are 20 of these. They're all different shapes, sizes, charge, hydrophobicity, and you build up a very, very long string of these that then fold into a big, complex machine that does something interesting. This all happens in the context of the cell. Depending how much biology you know, we get taught that the cell is a membrane inside which you have a soup of proteins and DNAs and sugars. This is broadly true, but the cell is a very highly structured environment. So without getting into all the detail, it's not worth listing through these, but just to convey, the cell comprises lots of very carefully structured, very tightly regulated machines, areas, subdomains, organelles. It is not just a bag of soup. And so one of the things that the virus has to do when it gets in is find its way through this, get the right parts into the right parts of this huge, enormously complicated factory. More computer-oriented people sometimes like to think about this in terms of information flow. Information starts in the nucleus or comes from outside the cell to be picked up by a lot of sensors on the cell surface and brought in. So information is constantly flowing through and around the cell, decisions are getting made, and the factory, if you like, is responding appropriately. So, cell as a self-maintaining information processing system for a computery crowd. DNA is the long-term storage, the source of truth. RNA carries the message or sometimes performs functions on its own, or both. Proteins are the machinery, more or less, and membranes control where things happen. Again, more or less. Lots of hand-waving here. Each of these sentences you could spend a career on and still not fully understand it. God knows I don't. So, hacks and espionage. We're going to talk about what a virus is, what it does when it gets to a cell, and what constraints under which they operate. So, viruses are not a factory. If the cell is a factory, a virus is not. They are a small set of instructions encoded in the viral genome and just enough machinery to get it into the cell and to take the cell over to make more copies of the virus. For a sense of scale, by the way, if a human cell is as big as the circus tent, a virus is somewhere between a grapefruit and a football, depending on the virus and depending how badly off my maths was. These are some examples of viruses. The point I want to make here just is that viruses are incredibly diverse. So we have, working from left to right, tobacco mosaic virus, which has an RNA genome and is a long rod. It's actually comprised of just one protein that stacks up and up and up and up in a spiral, like a spiral staircase, until it's long enough to contain the genome. Then we have adenovirus, which infects humans, and is a big, complex virus. It's quite big and chunky by virus standards. Then we have influenza virus. This is very different. It has a segmented RNA genome. So adenovirus has a double-stranded DNA genome. Influenza has, I think, 8 offhand, different RNA molecules that all need to be assembled together to form its genome. And these are surrounded by a layer of membrane that was stolen from the cell in which the influenza virus was made. Unlike the adenovirus, for example, which is just a protein coat. The adenovirus is a sort of hard protein shell. The influenza is a squishy membranous blob. And then on the far right is bacteriophage. Looks like an alien space lander, is great. These specialize in infecting bacteria. They're very different from human viruses, but we love to include them in presentations because they look really cool. In this talk, by the way, I'm going to be focusing on viruses that infect mammals, generally humans in particular. So all of these, although they're very different, they share similar constraints. Because they need to be small, they therefore have very limited genome size. That means a lot of things, so they can't bring their own complete replication machinery. They rely on hijacking a bunch of the cell's machinery and persuading it to make more copies of the virus. It has to have a way of getting into the correct cell because its machinery is set up specifically to hijack a certain kind of cell. It has to get to the right part of the factory inside the cell in order to manipulate those bits of machinery. It has to be able to, once it's there, take over what the cell is doing and persuade it, make copies of the virus instead of staying alive or instead of making mucus in your nose or whatever the virus is trying to stop the cell from doing. And it mustn't get caught too soon. Ideally, a virus would never be picked up by the immune system, but it's good enough if the virus can make copies of itself and spread before your immune system shuts it down. So how do we approach some of these problems? So the first one, data compression. Quickly, we're going to very quickly talk about having genes, which can carry a lot of traits. Basically, a gene is a stretch of DNA which encodes how to make a protein. We start off with what's called a promoter region, a regulatory region. This is a stretch of DNA with a correct sequence such that a bunch of different cellular proteins can land on it, recognize that sequence, and say, okay, this part of DNA is a signal that whatever's downstream needs to be copied into RNA to be made into a protein. And then you have the gene itself. We have exons and introns, which are not worth getting into too much, but we have the DNA sequence that encodes how to make the protein. There are edits that need to be made from this to remove what's called introns, which are short stretches of DNA which are not useful for making the protein. So they need to get snipped out. So we have a stretch of DNA. The proteins, the transcription complex lands on the promoter, chugs along the DNA, making an RNA molecule. Some modifications happen. Some more modifications happen, and the introns get spliced out, and we have a mature RNA molecule that contains just the sequence needed to make the protein. And then this gets, we say, translated into a protein. So this is a normal mammalian gene, normal human gene, and roughly how it gets from a gene to a protein. If we look at an AAV genome, so this is the genome from one of my favorite viruses. It's a very, very small virus. You probably have been infected with something like this before in your life and not known it. It would have been asymptomatic. This is a tiny, tiny virus, so it has a very compressed genome. Part of the way it does this is by getting around some of these constraints we've just talked about. So if we look at the top row, we have P5, P19, P40. These are different promoters. So these are the areas of DNA where the replication complex lands and says, oh, this is the start of a gene. I need to start making RNA from this. What you'll notice first of all is that the first promoter, called P5, sits outside the gene as normal. The next two, called P19 and P40, are actually inside the coding region. So the virus has managed to evolve protein coding regions, so DNA that contains the correct sequence to make a functional protein that it needs to replicate, that is also a correct DNA sequence such that some of the cell's replication machinery can land on it and say, okay, this is the start signal for something I need to make into RNA. This is sort of hugely difficult to do because you're meeting two very different sets of constraints. So okay, we've got promoters inside coding regions, quite tricky. We can also see that from this rep region, the 4 rows below it, we have rep 78, 68, 52, 40. These are all different proteins being made from the same coding region. And I talked about very briefly about the RNA maturing and bits of the introns being spliced out. The virus exploits this system and uses a combination of different start site from the first promoter and the internal promoter, and then different splicing strategies exploiting the cell's machinery to produce different sets of proteins from that. So we get 4 different proteins from one coding region. The VP1s do something very similar. What's really interesting is AAP and MAAP, so assembly activating protein, matrix associated accessory protein. Not getting into what they do, but this +1 here, these are in what's called a different reading frame from VP1. So they're doing something very special to squeeze more information into the same DNA sequence. So how does a DNA sequence encode a protein? A protein, we said, is made of amino acids. There are 20 amino acids. There are 4 letters of DNA, A, C, T, G. How do we get that? The answer is that they get read in triplets called codons. So we have our DNA strand at the top, gets copied into RNA, and then if you read in triplets, the UGG gets read as tryptophan, the UU gets read as phenylalanine, and so on. So these are sets of 3 letters. But how do we know which sets of 3? Why UGG and not GGU? So what we can have instead is alternate reading frames. So the row in red, each of these K, W, T, K, T, I, C along the top represents a different amino acid, reading one set of triplets. But if we shift into a different reading frame, we can see the blue letters, M, N, E, N, L. I think I've just failed my eye test, M, N, E, N, L, whatever the letters actually are encoding a different string of amino acids from the same codons. This works mostly, but it's hard to do. Like if you draw an analogy with English, this is our DNA sequence at the top, nice together. You can split it up in different ways, right? You can have a nice age to get her, or an ice age together. And so the protein encoding is sort of doing this trick. The analogy isn't perfect, but it mostly works. You have a specific sequence which, reading it slightly shifted one way or the other, encodes very different information. I will say this is quite a fun puzzle. If you get stuck in the queue for the bar later, coming up with these sentences is more interesting than I expected it to be. But like with English, you can get a few words. The few words strung together don't make a lot of sense. It's enormously difficult to write complete sentences or to have 2 different meaningful stories in the same string of letters. Enormously hard. But as you can see from the AAP and MAAP, these are very, very different proteins from anything else in the rest of the virus. Each of them does 2 or 3 different jobs, and the virus manages to encode them in the same string of letters, just one offset, as it does the VPs. It's a beautiful piece of squeezing lots of utility into a small amount of sequence. So another approach, this one is used by poliovirus, among many others, polyproteins. So I spoke about having a promoter driving a gene. Another way you can do this, have a promoter driving an enormous gene. You make one huge protein or polyprotein, and then that gets cut up progressively into smaller and smaller chunks, each of which is a functional protein, and many of the intermediates themselves are functional. What's particularly nice about this is that some of the subunits are proteases, they're called, which work to make subsequent cleavage events. So you get an initial cleavage into the big subunits, one or two of those act on other subunits to accelerate the cleavage to make different subunits. And this acts as a sort of a crude clock in the virus replication cycle. So over time, the pattern of available subunits changes. Some of those change the way in which it breaks itself down, and you get a shift from the genes that are needed early in the cycle to the genes that are needed late in the cycle, existing as proteins. It's a really, really beautiful system. So use of polyproteins. And I mention that because this is a lovely thing which coronaviruses do, including everyone's least favorite coronavirus. This combines these two ideas. So the coronaviruses all have what's called a slippery sequence. So the mRNA has a sequence in it which the machinery to translate it into protein does not cope with that well. And so some of the time, about 40% of the time, it accidentally makes a frame shift. So it goes from reading one set of triplets to the next set of triplets along, just one letter different. And this has the effect that if that frame shift does not happen, we get a shortish polyprotein. If the frame shift does happen on the slippery sequence, we get a polyprotein that's double the length. This happens about 30–40% of the time, depending which paper you read, exactly the experimental system, but a minority of the time. And that percentage is hugely important because the virus needs an exact ratio between the short polyprotein and the long polyprotein, which relies on the frame shift. And so it's been demonstrated that if you introduce very small mutations into this and that frame shift happens at a different rate, the virus just doesn't work well. And so this is a beautiful way that the virus can regulate its own production of viral proteins and control what information gets expressed from the genome by relying on the cell's machinery to just hit the slippery sequence. Some of the time it will slip, some of the time it won't, and that happens at exactly the right percentage to make the virus work perfectly. It's a fantastic system. So we've talked a little bit about information compression. I appreciate I'm going quite fast. How does the virus start to take over the cells? What does it do? So cells typically, when we're talking about replicating, have a bunch of different phases they go through. And it's more or less, a cell doesn't just sit there and decide, so, okay, I'm going to divide into, okay, now I'm going to divide into again. There are a bunch of very well-defined processes it goes through, which includes checking the external environment to say, am I getting signals telling me the body wants me to divide? And its internal environment saying, do I have enough energy to divide? Have I finished replicating my DNA? Are there any stress signals? Most of our cells, most of the time, are not in this cycle. They are what we say is quiescent, or we could say they're in G0 on this diagram. What viruses have to do is push the cells into a state mostly closer to S phase, which is where there's lots of protein production going, there's lots of DNA replication going, because that's the machinery the virus needs to hijack. And so viruses generally have to encode proteins which push the cells from a nice, content, quiescent state where it's just sitting there being part of your nose or part of your ear, into a state where it's not dividing, but it's getting ready to divide. So the virus can then insert its genome into that machinery to get copied. And as an example, adenovirus, another very nice example virus. Without getting too far into this cascade, it has these early genes called E1A, E1B. They have a lot of subunits, it's not worth getting into it. And they attack this on several fronts. So they get themselves involved in the DNA replication machinery. They sort of push themselves. They send some signals within the cell that starts to push it towards this S phase, where the cell suddenly wants to produce protein, wants to replicate DNA. And at the same time, it overcomes these blocks, which normally act as a sensor. If the cell feels itself going into S phase, and it doesn't have signals telling it this is okay, it will normally activate stress responses to say, I don't want to make protein, I don't want to make DNA, something's wrong. And if it gets pushed too far, the cell will shut itself down and sort of ultimately sort of commit suicide in a process called apoptosis. But adenovirus, among other viruses, attacks these systems, so it's simultaneously pushing the cells toward S phase and inhibiting P53 and BAX, which are involved in this cascade of shutting down the cell replication and going towards suicide. So it's forcing the cell forwards and then holding back its stress response. And fighting against the stress response is hugely important. This is way too complicated to get in. It's just to make the point, Our cells have been co-evolving with viruses for forever, for as long as there have been cells. Our cells are very finely tuned to try and fight against viruses doing exactly this to them. We have all sorts of molecular systems within our cells to detect hallmark traits, both of viruses specifically, and also of the cell being pushed into a state that it doesn't want to be in. For example, being pushed towards protein replication, being pushed towards DNA replication that the virus desperately needs. And every red box on this diagram, no, every green box on this diagram, I beg your pardon, is a viral mechanism that's been identified that's overcoming some part of the cell's response against viruses. There are huge networks of pathways of proteins interacting with proteins, making sugars, phosphorylating each other, and sending massive amounts of information all the way, all the time, all throughout the cell, detecting specific hallmarks of viral infection and triggering different systems to shut down protein synthesis, shut down DNA synthesis, send alarms out to the immune system to shut down the whole cell itself. And the viruses have evolved mechanisms to attack and shut down every single one of these. One of my personal favorites is called virus-associated RNA-1, VII-RNA, again from adenovirus. There's a system in your cell called PKR, which has evolved to sense the presence of double-stranded RNA, which is a very characteristic hallmark of RNA of virus infections. So PKR normally binds double-stranded RNA, and then it starts sending out signals all through the cell. We're infected, shut this down, shut that down, alert the immune system. It's one of the security guards of the cell. Virus-associated RNA-1 folds up into this beautiful structure, binds hard to PKR, but doesn't activate the alarm. So normally PKR would bind any double-stranded RNA, set off the alarm. This particular piece of double-stranded RNA, because of the shape it folds into, binds protein kinase R directly, stops the alarms going off, and allows the virus infection to proceed. And what it does simultaneously is part of it gets cut off, gets processed by part of a different part of the antiviral response within the cell. And that small part goes off, and the sequence of that part is important for shutting down other parts of the viral immune system. So this is a beautiful example of it's a gene within the virus that is expressed to RNA. Instead of the RNA making a protein, both its 3D structure that it folds into and the sequence of the RNA itself are important and play vital roles in interacting with proteins within the cell and shutting down the alarm system that would otherwise stop the cell, or that would otherwise make the cell stop the virus from replicating. A different alarm system is HLA, sometimes called MHC, depending where you're reading about it. The distinction is not that complicated, but not worth getting into. Our cells are by nature very paranoid. Because they've evolved to avoid being infected, all the time, every cell is sampling tiny bits of the protein inside itself at random. It chops out a tiny bit of that protein, it sticks it on an HLA molecule, and it goes through a processing process and displays it on the outside of itself. And then the idea is that all the cells are decorated with tiny, tiny, tiny bits of all the proteins that are being made within the cell at that time. The idea being that the immune system can come along, have a look, have a sniff. Is there something here which shouldn't be here? Has the cell shown me that it's got a virus protein inside? This is a beautiful, elegant system. And of course, viruses have evolved to attack and shut down every single part of it. This ends up being sort of quite a fun another fun arms race, because the viruses work hard to shut the system down because they don't want to be displayed on the surface of the cell for the immune system to find. The immune system, on its turn, has evolved a system where if a cell doesn't have any of this on the surface, it will just kill that cell because it assumes it's been infected. It can say the alarm system, the display system is not working on this cell. It must be because someone has shut it down. We're going to destroy that cell. And so you have this very delicate balance. The virus needs to inhibit this display system as much as possible to avoid the detection. But if it inhibits it completely, it goes too far, the immune system will just kill the cell anyway. Again, this is a subject you could spend your career on, understanding the details of the immunological response on how cells cope with viruses. But I find this sort of internal surveillance, the cell constantly checking within itself, here's a bit of me, is it fine? Here's a bit of me, is it fine? And the immune system constantly sniffing and tasting and saying, that's fine, no, you're infected, get the block. And the virus is fighting against this system. Yes, and so finally we get into you are part virus. So we're going to talk a tiny bit about retroviruses. Retroviruses have RNA genomes. They make their way into the cell. What's interesting about them is they contain the mechanisms for the RNA genome to get copied backwards into DNA. So we started talking about DNA, RNA, protein. The retrovirus instead started RNA, goes up to DNA, and gets itself incorporated into the cell's genome. And the cell just, okay, this information is in my genome, it must be fine, I'll use it to make copies of the virus. And that information stays in the DNA for the rest of the life of that cell. These can be very persistent infections. Yes, and so what happens if that cell is a progenitor cell that will go on to become either a sperm or an egg? And the answer is, the progeny from that have this same insertion in their DNA. This might be fine, it might be a problem for the infant, for the child. And if you look at our genome, our human genome is a graveyard of retroviruses that have made their way into our DNA, been silenced, their genomes have been fragmented, but they are still there. You can see huge quantities of retrovirus DNA that are still built into our genome. A lot of it is too fragmented and silenced to have any effect. It's just there, it doesn't seem to do anything. But others have become useful. So the famous example that everyone giving this kind of talk gives is syncytians. So when a retrovirus meets a cell, if you picture two soap bubbles coming together, they fuse and become one, and it releases the payload into the inside of the cell. This happens because of proteins called syncytians, which fuse membranes together and can fuse cells together. And the syncytians are essential in the formation of the placenta in humans now. At some point far back in our evolutionary history, a retrovirus got into one of our distant ancestors, hung around, and the retrovirus proteins have become essential in us becoming placental mammals. And there are all sorts of beautiful examples like this of how viral genes are necessary for us as humans to survive and for making us what we are. And then going very slightly in a different direction, lines, ALUs, and signs, oh my, have very distinct evolutionary origins. These are not old retroviruses. However, if you look in our DNA, you can find loads. I couldn't find a nice diagram with the human, but the mouse, you can find loads of this kind of structure. It looks extremely retrovirus-like. Out of our human DNA, it can express genes which include reverse transcriptase. So it gets expressed, and it has the machinery to make copies of itself and insert it in a different part of our DNA. So we have these elements which, in our own DNA, which can be expressed like a retrovirus, get itself copied back into DNA, and insert in a different part of our genome. So these are not viruses, they are just part of the human DNA. And what's fun about this is, if this is a complete retrovirus that can replicate on its own, this is a parasite of that. So a sign cannot on its own replicate, but if somewhere there is a line, which is the very virus-like structure, the sign can hijack its machinery to get itself copied. And something like 15–20% of the human genome is these, that over evolutionary history have been copying themselves and copying themselves and copying themselves all throughout our genome. The vast majority is inactive, but at the moment, we are all expressing at least fragments of these. And I think it's something like 1 in 20 births, something like that, has a new copy of something called ALU, which is very sign-like, and 1 in a few hundred births, that baby will have a new copy of line, because that event has happened in a sperm progenitor cell, that a bit of human DNA that looks very much like a virus has copied itself and hopped elsewhere. So all of this is to make the point that Viruses are extremely diverse. They have very different genome structures. They look very different. They behave in different ways. But they operate under very similar constraints. They're all trying to break into our cells, solve the same problems. They are tiny, so they use lots of information compression. And they need to overcome the immune system. And some of them integrate themselves into our genomes. This has happened a lot in the distant past. And a lot of the things that we do that make us human, that help us survive, are direct results of viruses integrating into our genomes, turning out to be useful, and hanging around for generations and generations. And then also making the point that a substantial chunk of our genome is very virus-like, expressing genes that don't look anything much like normal human genes and exist only to make copies of themselves and hop around within the virus genome, within the human genome, I'm sorry. So this is trying to make the point that we are all part virus one way or another. If you are the kind of person who likes reading about intricate systems and learning about sort of complicated ways that different systems can be broken and manipulated, I strongly encourage you to read up on molecular biology and viruses. This has been very fast, I apologize, but hopefully it's whet your appetite a little bit for what I think is a really fascinating subject. Thank you.